All right. Research talk number 2 for today afternoon from Narendra Dixit. Narendra is at Indian Institute of Science here in Bangalore, and he's in the Department of Chemical Engineering. But he does a lot of interesting work again that has relationships to evolutionary biology. So we're very happy to have him here. And he's going to talk about antibody responses after immunization. Also look up his other work to do with how dengue evolves and so on. I think it's very nice work. So look him up and catch him later.
Thank you very much, Deepa. Thanks also to Kavita for this very kind invitation. It's a great pleasure for me to be here and talk to you all about some of the work that we've been doing. I understand that I'm supposed to talk a little bit in terms of giving you examples of where evolution, perhaps, or population genetics plays out. And I'm going to try and do that with some of the work that we've been doing in the recent past. I was trying to think about what there might be to connect between the previous talk and mine. And I thought perhaps one could look at it this way. What Supratim was trying to see is what could give rise to substrates to begin evolution. And here we are talking of a process that happens in humans as we speak today. So it's a completely evolved process. And yet in this evolved process, you have evolution being selected, because there's evolution going on as we speak inside this evolved entity. So I thought that was quite interesting to see how once you have a substrate for evolution, that you might actually select for evolution eventually. I'll use the example of antibody responses to communicate some of these ideas. Let me begin by highlighting a couple of questions and then talk to you about the framework that we have used to think about them. The first is to look at the evolution of antibodies themselves. I'm going to try and see if I can motivate this by looking at HIV infection. What's plotted here on the left-hand side is the affinity that a particular antibody has for its target in an HIV-infected individual, and as this affinity evolves as a function of time. So you see here that the affinity grows, and this affinity is interesting. It's important, it's useful, because it decides how tightly this antibody binds to its target, and therefore decides the speed and the facility with which the virus is going to get cleared from the body through neutralization and other immune processes. So the larger this antibody affinity, the better it is. So you now look at this affinity evolving. This is the lineage of antibodies. So there are sequences that people have identified, mapped them onto phylogenetic trees and so on. So there's an ancestor here, and then this is the final evolved antibody. And you see that these affinities evolve over many orders of magnitude. Right? And then at some point, these affinities plateau. How this affinity evolves is something that people have understood. This plateau still remains a question. Now, if you look at many such realizations, many instantiations of this process, and look at this final affinity, right? And you plot them here. This is what is shown here on the right-hand side. And you see that this affinity lies in a certain range. And what is interesting is that this affinity is well below what could have been realized if this whole evolutionary process were to happen in vitro, right? And that's this number. And it's also much lower than the maximum affinities that have been realized between proteins. So if you take, for example, streptavidin and biotin, some proteins that bind very, very tightly, then their affinities are several orders of magnitude higher than the affinities that have been realized in vivo. So the questions that you can ask from observations like this antibody affinity for target, it increases with time. This process is called affinity maturation. How does this happen? And interestingly, that this affinity saturates well below maximum possible levels that are realizable in other systems. So what decides this affinity ceiling? Now, when it comes to HIV, most of you probably realize that this is an incurable disease. Many attempts are being made to see how one can bring about a cure. Of course, antiretroviral treatments are there. They control the virus levels, but do not get rid of it. So if you stop antiretroviral treatments, then the virus comes back up again. So of the many strategies that have been tried now to see if one can live with the virus controlled, but without using antiretroviral therapies lifelong, is to use antibodies. So antibody therapies are a strategy that are under active consideration. And by antibody therapy, these are sort of mimics of what one might have realized as plasma therapies during COVID. You take antibodies and put them in the body. This is a traditional treatment. It's been used to bring pathogen levels down swiftly so that the body gets some room to mount its immune response and so on. But even with these processes, HIV still has not been cured, but interesting observations happen that have implications for the evolution of these antibody affinities following this antibody treatments. And I'm going to talk about that as the next question that I'll pose, and then look at ways in which we think about this. So here is this business called passive immunization, which is the insertion of antibodies in HIV infected individuals. And this is an experiment that was done in a few years ago. The experiment is the following. Take individuals who are infected with HIV and you subject them to one injection of this antibody. This is supposed to target HIV. And now what you do is draw antibodies from these individuals, of course, before you do this infusion. And then you do the same drawing of the antibodies long after the administered antibodies are completely cleared from circulation. Now you look at the ability of these antibodies to neutralize the virus versus the ability of these antibodies to neutralize the virus. And here's the observation. This left panel is untreated controls, which means that this antibody was not administered. The right are people given this dose. And here there are two groups. This group is of individuals who are not on antiretroviral treatment. So they have high viral levels in their body. And these are individuals who were on antiretroviral treatment. So their virus levels were controlled. Each bar here is one individual. And what is shown here is a difference in the ability of these antibodies to neutralize the virus versus these antibodies. If you did not get this infusion, then it turns out there's hardly any difference. This is just noise. But if you were given this, then there is a significant improvement in the ability of these antibodies to neutralize the virus relative to these antibodies. And this, it turns out, is a lot lesser when you are on antiretroviral treatment, but it's still there. So now, these are endogenous antibodies because the administered antibodies are gone. Antibody infusions are supposed to control the virus. Here what's happening is that this antibody infusion is actually modulating the endogenous antibody response. So it's playing around with this process that causes the antibody affinities to evolve. So what you're seeing here is this passive immunization improves endogenous antibody responses in what has been called as the vaccinal effect, because you have some memory of this antibody infusion long after the antibodies themselves are cleared. And this improvement is actually better when individuals are not on ART. Right? So the question now is why? How does this happen? Right? So here are the questions that I'm going to try and see if I can address in the next few minutes. What determines the affinity ceiling? By what mechanism do external antibodies affect endogenous antibody production? And can this mechanism, if we identify it, be exploited to improve responses to immunization? So these are the questions. So let's see how to address them. For this, we must first go into the process that gives rise to this evolution of the antibody affinities. This process is called affinity maturation, as I said, and this happens in germinal centers. So these are structures that are temporarily assembled in our lymphoid organs following an infection. And it is here that B cells, which are the ones that produce these antibodies, get selected for their ability to target the particular pathogen that has triggered the formation of the germinal center in the first place. Here what you see is actually Darwinian evolution in fast forward mode. Right? So what happens here? This germinal center has a peculiar architecture. There is a light zone and then there is a dark zone, and these names are simply dependent on how densely packed these systems are, and when you look at them in a microscope, one side appears light and the other appears dark, and so on. In the light zone, there are follicular dendritic cells, and these follicular dendritic cells have on their surfaces receptors which can present antigen. This orange thing here is the antigen, complexed with antibodies. So the blue thing is the antibody there. The yellow are the receptors of the follicular dendritic cell. In the light zone are also B cells. Now these B cells have B cell receptors. These B cell receptors are transmembrane versions of the antibodies. These B cell receptors can latch on to these antibodies and extract them from this complex. When antigen acquisition happens, sorry, I'm just going to say one more thing here. This antigen acquisition now depends on the affinity of these B cell receptors for the antigen. So if I, for example, represent this antigen using some bit string, and I look at the matches between the receptors, which I could also represent using bit strings, with this antigen, then a filled box would be a match and an empty box would be no match. And so here you have somewhat of a better match of this receptor for this antigen than this receptor for the same antigen. So this B cell will acquire this antigen preferentially over this B cell. So when you acquire this, you get a survival signal. The B cell survives. If you don't, then the B cell dies. Right? This is already a layer of selection that is taking place here. Once you acquire this antigen, you get a second signal for survival from another cell inside the light zone called the T follicular helper cell. Now this, after you get a survival signal here, and I'll talk about what the survival signal is in a few minutes. Once you get this survival signal, then there is a fate decision that this cell makes. It experiences one of three decisions. For example, it could become a plasma cell, which means it gets out of this germinal center and becomes an antibody producing factory. So it produces many antibodies. These are essentially secreted versions of these B cell receptors. The other is that it could go back to the dark zone. Of course, some of it could also become memory, a small amount become memory B cells. You go back to the dark zone, and in the dark zone, you now see proliferation and mutation. A large portion of these selected B cells actually go back to the dark zone. During this proliferation and mutation process, the things that get mutated essentially are these antibody genes. So you have a large amount of mutation. These are among the most mutable regions of our genome. So you now produce a new collection of B cells which have altered B cell receptors. These B cells now come back to the light zone and they're subjected to the same selection process all over again. Right? So now if you have, for example, a B cell with a receptor that has an even higher binding affinity for this antigen than this one, then this is the one that's going to get selected. This one is going to die. And therefore, you now have a new plasma cell which has antibodies that have much higher affinities for this target antigen than the previous ones. So this is how affinity maturation takes place. This is the process. Right? So you're seeing now how this whole Darwinian evolution is taking place inside this germinal center. Now, these antibodies, there's an interesting process here. These antibodies, it turns out, can feed back into the germinal center. This is now a self correction, not correction, it's a control mechanism, if you will. These antibodies come back here and they compete for antigen with these antibodies that are already presenting antigen. So because these have now higher affinities, they preferentially take the place of these antibodies and present antigen. Now this makes the task for these B cells tougher because they have to pull antigen from something that is more tightly bound to the presenting antibody. So the selection becomes more stringent. Our hypothesis is that these external antibodies do exactly that. Because these antibodies are feeding back, the external antibodies can also come in. They increase the selection stringency here because the external antibodies, which are used as passive immunization, are usually designed to have high affinities for their targets. They increase the selection stringency here and therefore cause affinities of these antibodies that subsequently get produced to be much higher than otherwise. What's the actual process there? So antigen presentation by passive antibody is really what goes on. This is an expanded view of the follicular dendritic cell, the receptor of the follicular dendritic cell on which this immune complex or the antigen antibody complex is presented. Passive antibodies could come there, and this is just a cartoon. This could happen elsewhere in the system as well. But then they are going to compete for this antigen with this presenting antibody. And if they have much higher affinities, they're going to snatch them off and themselves serve to present this antibody. And if you can now tune the affinity of these antibodies, they could be weak or they could be very powerful, tightly binding antibodies. And then this now makes the extraction of antigen by the B cells much tougher. I have a B cell here, which was previously extracting antibodies from from this antibody. Now it instead has to extract antigen from this antibody, which is a much tighter binding antibody. So you get selection now of B cells that only have receptors that are even more attracted or have much higher affinities for this antigen than this antibody. Of course, the downside is that you're going to get a lot fewer of them, but then that's how you modulate now the selection process through these administered antibodies.
The previous data that you showed, is there evidence that these modulated affinity is higher than the Like what you're showing here?
But the explicit affinity measurements are not there, but neutralization is usually a very good surrogate for affinity. So they do see increased neutralization.
Bigger than what the passive antibody.
Yes, yes. Okay, so now to quantify this whole, yes. As I said, light and dark zone is just it's a nomenclature that has become fixed in the field based on microscopy. In the early days, when people did microscopy of these things, they found that there was a larger density of cells in the dark zone, and so they appeared dark when you did light microscopy, and that's why it's called a dark zone. There's no other real reason. Interestingly, that the cells would go back into the light zone was something that was first predicted through theory by some theoretical immunologists who were asking what's the optimal way for producing antibodies. And then later on, when imaging became more sophisticated, it was discovered to be true. All right, so we set up stochastic simulations of this process. This is really a Wright-Fisher type simulation that one can do because you have generation after generation of these B cells coming in from the dark zone to the light zone, undergoing mutation, and then being subjected to selection. And that's, of course, the selection is more sophisticated because there are 2 steps involved, but really that's all there is. And so you can set this up. So we set up these simulations. We've picked a fixed population of these B cells in the light zone, initially with B cell receptors that had 4 affinities for this particular antigen. We assume the antigen was fixed. You can also make this more sophisticated by letting the antigen evolve, which is what really happens in situations like HIV and so on. And then you have these antigen being presented. Each B cell gets a certain number of chances to pick the antigen from this antibody complex. And when it does, it gets a survival signal. And in this particular case, because it's all affinity driven, we had a formalism for deciding what the probability was of this B cell successfully pulling out this antigen from this presenting antibody. And we said that that was basically proportional to the affinity difference between this B cell receptor and the presenting antibody for the antigen. And then you had this B cell now coming for T follicular cell helper cell help. And this is an interesting process. It turns out that this follicular helper cell simultaneously binds to many B cells and preferentially provides signals to the B cell that presents the most antigen among the B cells that have simultaneously bound this particular T follicular helper cell. So to model this, we said, in principle, more antigen, the more the likelihood of you getting the survival signal, but more antigen relative to the peers that are competing for that antigen. So if you had antigen, the probability that you got this T cell help was therefore proportional to the amount of antigen you extracted, which is the number of successful attempts you made in this process, minus the average antigen that other B cells in the milieu had. So now you could go, and then once you had the survival signal, 5% of the time you became a plasma cell, 5% of the time a memory B cell, and 90% of the time you went back to the dark zone, got your B cell receptors mutated, and then came back for selection. So this was the process. Okay, and then of course you had antibodies feeding back. So this is just to give you some details. We assume that you had a bit string of 3, could make it much larger. 1000 B cells are typical of a germinal center. 8 attempts per B cell, 1 attempt at getting T follicular helper cell help, 2 rounds of proliferation in the dark zone per B cell, 10% mutation. This is also realistic based on the hypermutation rates, as they are called, of these antibody genes. 12 hours of 1 generation. Antibodies we assume were not limiting because they're passively administered antibodies. They're usually around in copious amounts. Antibody feedback, we restricted it to the past 3 generations, which was also reasonable. And then we assumed that these passive antibodies would dominate antigen presentation in the germinal center until endogenous antibodies had affinities that were larger than these passively administered antibodies, after which natural affinity maturation would take over. So we now set this up and ran these simulations. Here are some results. This is the number of B cells in the germinal center as a function of time following the start of this germinal center reaction that have different affinities for this particular target antigen. Low affinity, intermediate affinity, and high affinity. So you start out with a lot of low affinity B cells. These numbers decline, of course, because you produce these, because of mutations, the intermediate affinity antibodies, which win over in the competition, and then eventually give rise to these high affinity antibodies or B cells, and these then reach some sort of mutation selection balance, if you will. That's where the Jamal center reaction sort of ends. Now you ask what happens if you had passively administered antibodies of low affinity, right, in the milieu now. You can repeat this, and low affinity antibodies are not very interesting because these are already being produced by these B cells. So then not a whole lot of change that happens. But if you now put in intermediate and high affinity antibody, then you see significant changes in the evolutionary process. Two major changes happen. One is that the rate of evolution increases, right? Because you're increasing the selection stringency. And then the intermediate affinity B cells are suppressed. So you quickly shift over to these high affinity B cells. So in effect, you're increasing the speed of evolution, and then you're also tilting the evolution in favor of these higher affinity B cells. Now, some of these processes, are they real? Are they seen in experiments? So we looked at some experiments where some of these evolutionary processes have been observed. One of the observations that we made, and which is also seen in some experiments, is this business of the quality-quantity trade-off. Right? So now if I now repeat this evolutionary process as I did earlier with low, intermediate or high affinity passively administered antibodies, two things happen. As I increase the antibody affinity, administered antibody affinity, the output, the actual absolute amount of antibodies that are produced, they go down. And this is because you're selecting for, you've made selections very stringent. So fewer B cells are getting selected, but they have much higher affinity for the targets. So correspondingly, you see antibody affinities actually increasing, right? So this is your quality-quantity trade-off. So you lose quantity, but you improve on quality as you increase the affinity of the passively administered antibodies. So if I were to quantify this by looking at a snapshot of what happens, let's say 2 days after this evolutionary process has begun, you'll see the number of cells dying goes up. The population of the surviving Jamal center B cells goes down. The number of cells that are getting selected or being let out as plasma cells goes down, but the affinity of the antibodies that are being produced goes up. This is exactly what was seen in a mouse experiment, which did exactly this. They administered low, intermediate, and high affinity antibodies and observed the amount of cell death, the size of the Jamal centers, the number of plasma blasts or the cells producing antibodies that are secreted, and the quality of the antibodies. Yes.
In the previous plot as well as here, maybe you see much more variability in the affinity of the high affinity ones.
Yeah? Yes.
When you administer a high affinity and
this one, right? Yeah, it's a very good observation. This happens because when you have very high affinity antibodies administered, you can actually get a lot of the Jamal center just dying out because your selection is so stringent, some of them just don't survive. So you see this much larger variability there. Okay, another experiment. If selection stringency is large, then you might imagine that if I had a lot of antigen available, I allow more chances to these B cells to try and pluck these, to extract this antigen. Then I'm going to effectively reduce the selection stringency. Right? Would that then alleviate this problem, this trade-off? So this is an interesting experiment that was done, somebody else, of course, where they took, again, in this case now they're administering antigen. This is actual vaccination, sort of passive immunization. But then they had a certain protocol for administration of this antigen. The total amount of antigen that was administered was constant, but in one case, they administered it in exponentially increasing amounts per day. In another case, exponentially decreasing amounts, and in the third case, a constant amount throughout, right? Over a period of a week. What should happen? And here's what they found experimentally, that if they looked at the antibodies that were produced, in this particular case, they had the best antibody response, exponentially increasing antigen dosage. In this particular case, the worst, and this was somewhat intermediate. Right? We could recapitulate this. We ran our simulations, subjected them to the same antigen administration profiles, and in our case, what this antigen really does is increases less. If you increase antigen levels, it increases the number of attempts that each B cell has to extract antigen, because in the Jamal center now you have a lot more antigen available. Mimic this, and then you find that indeed this is the output of the Jamal center, that this is far better, right? The exponentially increasing one is far better than the constant one, which in turn is far better than the exponentially decreasing one. Why does this work out? And this is just another way of presenting the same result. Why does this happen? So as you increase, if you're beginning the evolutionary process here, now you produce B cells with higher affinities for the targets. But then if you now allow for more and more antigen being available, you let these B cells continuously improve and not die. On the other hand, here, as you increase the B cell, the affinity of the B cells as they increase, if you now decrease the antigen that is available, then you select far fewer of these B cells because there's just so much less of the antigen. So arrest the evolutionary process much sooner than otherwise. So this is an interesting way of recognizing that this quality quantity trade-off exists and that this antigen availability is a handle to tune this trade-off. So with these two observations, you can understand this experiment, right? So what exactly happens here? The increase that you see here is, of course, because in the germinal center, you now have selection stringency improved, and therefore you select for B cells that are much better at binding to these particular antigens, and therefore there's increased neutralization. And of course, why do you see this being better than this? Because there's a lot more antigen here, right? Sorry?
So using selection stringency. So do you mean selection coefficient is higher? Is that what you mean? Or?
No, it's the selection, by selection coefficient, you're talking of the difference in the fitness between the higher and
yeah, what do you mean by stringent selection?
In this case, it's just that the probability that you select, that you are able to extract antigen is much lower when you have higher affinity antibodies presenting the antigen, right? So you could be right. I have not thought about it in those very words, but in this case, let's say you have an on-off process in the sense that if your affinities of the B cells, let's say or you can think of a sigmoidal process, are too low, then you're not going to pull antigen and you're going to die. If they are higher than a certain threshold, then you're likely to extract antigen and you survive. This threshold moves. Yes.
So, is there a role for the memory B cells? The 5%
Yeah,
go back in this pool or produce others that go back?
Yeah, in a running germinal center, there's not so much of a role, but I will show you an example where there is a role for those things. Yeah. Right. So the difference here is what? High antigen, low antigen. Right. So we understand this a little bit at least. So then we said, if this process is what is going on, are there other examples where some of this works out? And more importantly, can we now use this to, for example, think about what's the optimal dosing protocol and so on? Normally, these passive immunization strategies use 3 doses. So there's dose 1, dose 2, and dose 3, and then they analyze responses after 2 weeks, sometimes a month, and so on. So we said we have the system or the framework for us to analyze what might happen if we did all of this. This is not done experimentally, but if we did this, then what would be the optimal sort of dosing protocol if I had, let's say, at my disposal, these different antibodies with different affinities for the particular target antigen. So you, of course, get 27 different combinations. We can run all of them and see what really happens. And it turns out the outcome is different depending on how much antigen there is in the germinal center. Low antigen, medium, and high, and you see different outcomes. So in the low antigen scenario, you find that the combination where you only administered low affinity antibodies is the one that produces the best response. Whereas if you had a lot of antigen, then the protocol that has the highest affinity antibodies used all the time gives you the best response. And this is again related to the quality quantity trade-off. Here, if you had very high affinity antibodies used, nothing would get selected because there's too much selection stringency, right? And here it's the opposite. And here you're able to break this quality quantity trade-off by having a lot of antigen available, and then higher affinity antibodies give you a much better response. This is interesting because the level of antigen that is present in the germinal centers could be related to the viral levels that people have in their bodies. And it turns out that this what is called the set point viral load, the viral load that people have in their bodies for many decades before they see symptoms manifesting, can vary over several orders of magnitude across individuals. So if that really translates into differences in the level of antigen that is being presented in the germinal centers, then the optimal protocols might actually be different for different individuals, and they might be tailorable. But that's something that I'm speculating on. Just that that's an interesting prediction that comes out of all of this, right? So you can now decide what is the optimum sequence, and it turns out if you have low antigen availability, then low affinity antibodies are the best ones, if you have the highest antigen availability, then you might have the liberty of using high affinity antibodies and producing both higher affinity responses and higher magnitudes of those responses. Okay, a couple of other asides, and then I will end this whole thing. Here's an interesting observation that we made related to COVID vaccination. And many of you may realize when the Oxford vaccines were being given out, that there was this debate about whether one should use low doses to begin with, high doses, or whether one should spread the two doses apart a lot more or a lot less, and so on. The reason this debate came about is that I think because of some technical error, there was a group of individuals in when this Oxford trial was being conducted who were given half the prescribed dose. The first dose that they were administered was 50% of the normal. And then the second dose they got corrected because somebody realized that there was an error. But when these individuals were followed, it turned out that they had a much better antibody response than the ones who were given the normal dose, right? And then they say, okay, maybe there is something going on here. And then people then did systematic studies and with other vaccines also. And in general, this notion came about that if you had lower doses of the first dose and then normal or higher doses of the second one, you tended to mount a better antibody response than if you had both doses being the same. And the same was also true if you spread the two doses apart. So now you can understand this from this framework. What happens if you have low doses to begin with, then your selection stringency is much higher inside the germinal centers. You tend to select for B cells that have much higher binding affinities for the target antigen. Unfortunately, you select a lot fewer of them than if you had a lot more of the antigen available. Right? But then you come with a second dose, which has a lot of antigen. So you allow now for these few selected B cells, which have higher affinities for the targets, to expand, to grow. And therefore, you overall produce a much better antibody response than if you had both the doses being the same. And this also is true if you increase the gap between the two doses, because as your affinity maturation progresses, selection stringency becomes tighter and tighter, and then you eventually select for B cells that have very high affinities. If you, on the other hand, came in with a second dose much sooner, then you would proliferate, you cause the cells that were selected until that time, which could be much lower affinities than if you waited longer, to expand. So just simple simulation that show you the same thing. This is what happens with the first dose. This is the average GC B cell affinity, and this is the overall output. First dose, second dose, and antibodies rise, and then when you come in with the second dose, there's of course this affinity maturation process that goes on. And in this case, we looked at two cases of both standard doses, low dose followed by standard dose. And in this case, we asked what could seed the germinal center after the second dose, right? It could be the memory cells that were produced from the first germinal center, or it could be the continuation of a running germinal center. And in both cases, you find that if you use the low dose versus the standard dose, you got better overall responses than if you used standard doses alone, right? This is just the representation of the same thing. This is where you had low dose followed by standard dose. That was better than if you had both standard doses, right? And in this case, if you continued the GC instead of seeding the GC with memory cells from the past germinal center, you again got better responses if you started out with a low dose, right? So this is just telling you that if this framework were true, if you account for many more germinal centers, the diversity of the responses, etc., there might be a way in which you can use these ideas to begin to tailor the doses to produce optimal responses. Okay, last thing that I'll tell you, where's the affinity ceiling coming from, right? It turns out that there are many hypotheses. This is not a settled problem. And what I'm presenting to you is also something that I would call a hypothesis. It's not over yet. So this is the architecture that's going on where the antigen acquisition is taking place. B cells, when they latch on to this antigen, they actually exert a mechanical pulling force, the pull on the antigen. And in this process, there's this chain of protein complexes that is getting tugged. You have here the FDC membrane onto which is attached this immune complex. This is an antibody, the presenting antibody. That in turn is attached to the antigen. And this is now the B cell receptor. And this is anchored in the B cell membrane. And this B cell is now undergoing cytoskeletal changes and pulling on this. It's pulling on this, and therefore it's trying to extract this. The question is, this extraction ends when this chain breaks. Where does the chain break? it turns out if you put in all the numbers, and it's a very simple argument, initially, when your B cell receptors have low affinity for the antigen, it's these bonds that are a lot weaker. This antibody is also produced by some other B cell. Everything is low affinity. So these affinities are a lot weaker than the affinity. These are now fixed regions. These regions don't change. It's these regions that evolve during this affinity maturation process. These bonds are fixed, right? So these affinities don't change very much. These are weaker. So initially these bonds break. But as these antibodies become tighter and tighter binding, eventually this becomes weaker, right? And this breaks. And when this bond starts to break, then this selection process ends. Because no matter what you do here, you're always going to break here. There's no advantage now for mutating anymore here. This is our hypothesis of how this affinity ceiling is being realized, and it's a lot lower than what could have been realized if your selection did not involve these extraneous bonds. If you put in the numbers, it turns out these numbers come very close to the ceilings that have been observed in experiments. Of course, it has to be proven by performing mutations and so on, but it might tell you that if you, for example, mutate this somehow, antibody engineering is possible. For this affinity to be a lot higher, then your ceiling might actually go up, and you might end up with a Jamal center reaction that culminates with a lot higher affinity B cells than what is currently being observed. All right, so I'm going to stop here and summarize by saying that there is this quality quantity trade-off that constrains the Jamal center reaction. The affinity of the passively administered antibodies, as well as their antigen availability in the Jamal center, can tune this trade-off and therefore engineer this Jamal center reaction. And just for a sort of speculative ending, the weakest sort of link hypothesis may underlie this affinity ceiling that's been observed. We thank students and postdocs who did all of the work and sources of funding, and thank you all for your attention.
So if this weakest link hypothesis is true, you'd expect to see different affinity ceilings depending on that weakest link affinity that exists. Is there data to support that?
No, so that, as I said, those are not really varying. These are.
So those don't vary even across different receptors.
I mean, them across different. No, these are exactly. These are constant receptors. Of course, your genes will be different, but they are not subject to the evolution that you're seeing inside the Jamal centers. Yeah. So there may be some variation, but not because of this evolutionary process. Yes.
Why is evolution showing up that not passive?
Sorry, I could not hear that. Could you please repeat?
So, like, why is evolution not taking place when you didn't administer the passive, like, antibodies from outside?
Without passive immunization, why is there no evolution?
There is.
No, but you show in your first slide that there is data that when you didn't administer the antibodies, in first case and in second case,
Oh, no. So there are two different problems. In the very first slide, I showed you that the antibody affinity goes up and then hits a ceiling, right? In the second slide, where I talked to you about the evolution with and without passive administration, I'm already talking to you about a situation where you've hit the ceiling, right? When you're already at the ceiling, you have no more evolution is taking place. And after that, if you administer antibodies passively, things do change. Hi, yes. Yes, indeed, that's a great question, and we have actually speculated on that in this essay of ours. And it's really that there could, if you did not have this limit, perhaps you might end up in runaway evolution, which might trigger autoimmune disorders and so on. So there's that angle to it, but it still remains a speculation because we haven't really worked the whole thing out carefully. Again, it's a great question, and there is some variation in natural variation, and in fact, we were thinking that people who have these ceilings being much larger could be more prone to autoimmune disorders, although they might also have much stronger immune responses to some pathogens. In other settings where people have looked at T cells, this coupling between stronger responses and greater autoimmune disorders has been observed.
Right, we'll break now for coffee. Thank you very
Indeed.
much.
Thank you.